Yeah. That's pretty easy. That's like Yeah. Yeah.
That's kind of applied to bro.
I was gonna do, my plan was to do direct on except for now. I was gonna take it in the summer, but after talking to the advisor, for one, it's too late. So I think I'll start like spur of the moment. Yeah. Yeah. I live in New York. It's not Jefferson City. It's about 45-minute drive here. I'm technically closer to Morristown campus, but it's a couple people and with my brother did not able to be. Like you would finish the year at one, and I haven't got work on there, so I got home.
You ought to be next door. It's even worse.
Oh, really?
Yeah,
Yeah, I'll stay here.
it's much shorter.
Can you control it right now? I know some places can and some places can't.
In theory, if I play with that, it works. But
But
Communication,
as far as you know,
there's
not really.
only two listeners.
I really.
Yeah, everybody else like calling.
So you're good.
It's been so many years since I went back.
Yeah, that's what happens with my high school, is that like some teachers have like germicides, but like change it. But they really couldn't change
Yeah, it's all okay.
Right, yeah. I'm 26, so it's been a little bit. I tested out of everything. Really well. I did a lot of studying. Yeah. Well, the reading and writing, like, I've always been that thing. So when I was taking the practices and I was doing better at the reading and writing, because there's just like the graphic and stuff, and then it's been so long since I did it. Like the nights before the test, I studied up on just a couple things I couldn't remember. And really what I did, those things were just test.
So I know. It's like, so that would have been such a mal.
I learned how to do it. I just bought this one yesterday from somebody that works in the theater. I have 3 lifelong spouses. Like, 2 of them want to turn on, and I won't get rid of them. I just don't know what's on it. I figure eventually I want to get a yeah, yeah. Yeah. And like I have one, it'll turn on, it's fine. But it has a CD drive or it had a CD drive. I have 2 kids, so I still have it. It's just not the same. But I don't want to get rid of it because I have to like keep the cars, big fingers, see anything like my kid. I figure my kids would really like play on it. Play games. Well, but I didn't have to CD drive because they messed up. Well, my kids have it. I was actually able to download. My kids were playing it, and I'm sitting there watching.
Okay, let's go ahead and get started now so we can get done. And we are going to take a look. We're going to open up our first lab here. Just a few things of information on the lab course. The AP1 Lab is a 1-credit course. We meet one day a week. I have the same lab on Tuesday, Wednesday, and Thursday. Don't come on Monday with the AP2s. So Tuesday, Wednesday, and Thursday, it's exactly the same lab. So if you have a conflict on scheduling, if you need to miss like the Thursday lab, you can come in on Tuesday or Wednesday. Of course, they're always going to be recorded. And I'm going to have recordings of all the labs. So you can, if you have to miss, you can't make it up or come in early, go ahead and watch it. But it'll be the exact same lab each week. So now, having said that, well, in lecture, our dates for exams are pretty flexible because I don't know when I want to get, finish a chapter, finish a unit, whatever. In here, we keep them pretty well set in concrete. The dates are very set because we have to finish up a unit because the next one has to start because we don't have enough time really. Let's just hope we don't have any weather days or anything like that, or the school closes, because there's no time built into the schedule to get everything done we have to cover. The schedule for the lab is found in eLearn under the content section for this particular class. It's BIOL 2011 H13. So if you click in there and look at content, you'll see the lab schedule. There's also a copy posted over there on the bulletin board, and there's a copy on the bulletin board at the other end of the hall because they don't give us bulletin boards outside down here. Anyway, those dates are pretty well set. What we're going to do now, there is a little bit of flexibility there, and I can move things around a little bit. So, but if you have a problem, you can't make your class, then try and come in one of the other ones. I've already told the Tuesday and Wednesday classes that there's some flexibility there for them too. They can, I'm the only one teaching A&P, so I can pretty much decide how I want to do it. So it doesn't bother me if people need to float around. Now, as I said in lecture, if something happens, things always happen. Okay, people always get sick, cars break down, kids get sick, you name it. I've heard, I've done, this is my 19th year at Walter State, and I've gone through every experience that students can have, and it all works out. I will do everything I can to help you out on a situation so that you don't have to stress over, thank you, over the class, because life has a nasty habit of getting in the way of what we want to do. So if something happens, just let me know. If you miss a class, if you miss a test, it's okay. And this applies to lecture too. It's not the end of the world. It just, it happens, so we go on, I'll fix it. I'll give you more time. I'll let you take it later, whatever, as long as it's a reasonable excuse, but about 10 years ago, I had somebody say, I couldn't get ready for my test because I was in line at GameStop on Monday night for the release at midnight. And I thought, I didn't get home till 2 o'clock in the morning. That's not a reasonable excuse, Because one year I was actually with my son in line at GameStop at midnight waiting for a release for him. And, so it's, it's just, I'm pretty reasonable. I think I actually was sympathetic because I'd been through that experience too. So, but I mean, things are going to happen. I'm not going to be I'm not going to get any heartburn over it. I'll help you however I can. So same thing applies to lecture, same thing applies to lab. Now, in here we have 4 exams, 4 lab exams. They are, they stand by themselves. There is no final exam in lab. I say that on August 25th. I know I'll say it again on November 25th or whatever towards the end of the semester. Because it just seems like as we approach exams, everybody seems to think, well, we got to have a lab exam too. Y'all don't want a lab exam. I don't want a lab exam. So there is no lab exam. You take test 1, you're done with it. You take test 2, you're done with it. You take test 3, you're done with it. And test 4, you're done with it. To look forward to something in AP2, there's only 3 lab exams. And I'll tell you that the AP2 lab, well, they'll be done the Monday before Thanksgiving. So we have less content to cover in there. We will be very busy. You have 4 exams. I urge you to prepare for the exams. Many people don't take the lab exam seriously, and they discover there we go. They discovered that we were serious about the questions that things you're gonna have to know. Now, and so people, will do very badly on the first exam. That often works as a wake-up call. And I've had lots of students who've done badly in the first exam turn around and do very well the rest of the semester, because there's 4 exams. You can afford to, you can sort of afford to get burned on the first exam if it shows you that we are very serious about the content you have to master. Now, having said that, also, I gotta stop saying that too. Having said that, the bone test is the second test. The bone test, I will give you 4 weeks to get ready for. You'll need it to memorize all the bone parts. So, and that's, while this first test doesn't have a whole lot of practical applications as far as I can see, as far as the microscope stuff goes, but there is some. Knowing all the bones of the body, knowing a fair amount of muscles of the body, knowing the structures of the brain and the eyeball, those all have good applications. It's probably why people do much better on the second, third, and fourth exam. Now, the other thing to remember is that this class is not going to get you into any healthcare program. This class is not going to get you into a healthcare program because nobody in healthcare looks at the lab grades. The only grade that's important is the lecture grade. So you need to do as well as you can on in lecture. You have to strive for that A or a B. I'll also tell you that nobody gets in with a C, even though the catalogs say you can get admission with a C. Very rarely does that happen, if ever. So, so you want to get an A or a B, and you really would rather get an A in lecture and a B in lab. The B in lab's not going to hurt you. The lab grade is 1 credit hour. It has a minimal impact on your overall GPA. An A in lab will raise your GPA probably like 1/100th of a point. A B in lab will drop your GPA by a 0.05 or 0.005, whatever. It's a minimal drop. Even a C you're not going to, it's going to be at least in the hundreds column. You might go from a 3.55 to a 3.54, if that. That's not going to hurt you. The only impact that the lab grade has is on your GPA. And the GPA is used to help calculate your points totals for admission into healthcare. The healthcare programs use points. They give you points for everything. And I know in nursing, they will take your GPA and, let's say it's 3.6 and multiply by 1,000. So now you have 3,600 points. If you have 3,600 points, if your GPA is 3.65, you have 3,650 points. If it's 3,645 points, it's not going to make a whole lot of difference. So the lab grade doesn't have a lot of impact either way. So put your focus on lecture. So put your focus on lecture because this is the most important class you're taking. You know, it doesn't do any good to get a great grade in psychology. And Dr. Rollinson would agree with me. If you're going, excuse me, if you're going into healthcare and you get an A from him and you get a C in here, that's not going to help you. You're not going to get into a healthcare program with an A in psychology and a C in A&P. Yeah. If you got a B in A&P, you got a better chance. You get an A in A&P, you got a great chance. I'm just saying that I know everybody tells you that their class is the most important class you got to take, and you know that's from their perspective. I'm just telling you from going into healthcare. If you want to get into healthcare, you can get into healthcare from my classes if you get an A or B. There's no other way around it. So I'll give you everything you need to succeed. And I'll bend over to make sure you have those chances. But you got to, you got to work with me, but I will help you. There, admission into any of the healthcare programs is all totally objective. It's all based on points. You want to earn those points. You get points for your GPA. You get points for A's. You earn an A in A&P1, you get 500 points towards your point total. This is for the nursing program. I'll use that as an example. If you get a B, you get 250. You take AP2, it's again 500 and 250. You take micro, there's another 500 or 250. If you knock out all your general eds, you get points for that. You get points for your overall GPA. You get points if you take the HESI, the HESI exam. You get points if you're only a year or so out of your not essay. What's thank you. I lived in Virginia, so we were SATs. So yes. So if you on your standardized test, you get points for that if you scored high enough. So all these things contribute to how you're going to earn your points. So you got to do well. Okay. Enough. Get off my little soapbox and stop talking about that. I don't want to scare anybody. Lots of people go through here and do quite well. I usually fill up, they have 30 slots, I think, in nursing here in Sevierville. And I usually put about 90% of my students in there and the rest come from the other campuses. So if you're in my class, you're going to have a real good shot at getting in because I'm going to make you work and get your grade and get you in. So, and that applies to surge tech or any of the OTA or respiratory or radiology, any of the programs. ETSU down the road has 4 slots for radiology, and usually 3 of them are students of mine. So I send a lot on. I'll get you there if you do well. So, questions on anything? Question?
Thank you for that. Well, this, okay, so I've been debating between nursing and paramedic, and I know that this will help me with my nursing, but will this class also help me with my paramedic courses if I decide to do paramedic?
Oh yeah, because anatomy and physiology is a major component of what the paramedics teach too.
Yeah, I just didn't know if I would have to take it with them or if this would count as like credit for that.
That depends on, you'd have to talk to the paramedic people themselves.
Okay.
But I've had lots of paramedics take this class too.
Okay.
So, and so it does give you some flexibility there to decide which way you want to go.
Yeah.
So it was a good question. Now, I've had lots of paramedics in the class over the years where they decide they want to go on into, some sort of advanced healthcare type approach. So anything that, yeah.
So the main exams, like more practical or just like on paper memorization?
Oh, the lab exams? Well, the bone exam will likely be a practical exam where I have a bone laying out on the table or attached to the skeleton, and I'll ask you what this is. or I'll ask you what of these processes, these bumps are on the bone. So that you can identify that, because that's a real practical application in any career you go into down the road in healthcare, being able to know the bone parts. And then the other big lab exam that's a practical based is the muscle exam, where you learn. Fortunately, there's 206 bones, but there's only 60 muscles we look at, so it's a lot easier. And I'll tell you, once you learn the bones, you never forget them. And it's a whole lot easier to learn the muscles if you know the bones. So those two are going to be practical based. Lab test 1 is going to be multiple choice. Lab test 4 is usually multiple choice. I will give you, for the practical exam, I will give you a word bank so you can go through there. And all you have to do is mark down the right letter, mainly because I got tired of trying to read spelling. Somebody, where it just, what is that? I don't know. I'll give you credit, but I don't know what it is. So if it looks anywhere close to it. So it will, and I'm actually lazy. It's easier for me to grade if I'm just checking off a letter or a number, as opposed to trying to read the words. So, however, I will say that many of the words are often very amusing, and we actually used to keep a list of all the words. Professor that's no longer here used to keep he called it the wall of shame, and he would write down these bizarre words he got. I'm glad he doesn't do that anymore, but it was still funny. So, but for the life of me, I can't remember any of the terms, at least not right now. So anyway, so yes. The two in the middle, the muscles and bones, will be practicals. The two in the ends will be multiple choice. But even the practicals will have the word bank. So you at least, you still got to know it, but it's a whole lot easier if you know it and say, oh, there it is, and there's the name of it, as opposed to spelling out sternocleidomastoid for a muscle. And just, oh, there it is. That's it. Mark it down. So. Other questions? Okay, let's see if anything else. Everything, the syllabus is in the content section. There is the lab schedules in the content section. Grading is 90 to 100, 80 to 89, just like it is in lecture. There are 400 possible points in here. You just need to get 360 points out of 400 to get an A. You need to get 320 out of 400 to get a B. It's as simple as that, so there is no extra credit. In the lab, we're not allowed to, that's a division policy. So we won't have anything for that. I am very flexible on making up exams if things happen, and things will happen over the course of the semester. So but always let me know ahead of time, or let me know as soon as possible, if something's going on that I can help you with. Let's see. Fire drills. If we have a fire drill or if the fire alarm goes off, we go out into the soccer fields. Now that the building is finally finished over there. The other thing is, if we do have a fire alarm, take everything with you. Don't leave it behind. Because if it's for real, you won't get back in. If it's not, then you're still delayed. And the only reason I emphasized take everything with you was about 4 years ago, I was in Morristown and they had a gas leak in the natural science building, and they evacuated the building. And some people insisted on telling their students to leave everything and get out of the building as quick as possible. And I can understand that, because when you're dealing with a gas leak, the last thing you want is a spark. However, they evacuated the building, they locked the building, and everybody that was outside, if they didn't have their keys or their books or their computers or their coats or anything, had no way to get them. And the college had to bus everybody home. So, and that would happen here too. You'd be taken care of, but can you imagine the inconvenience of all that? So, if nothing else, take your phone and your keys with you. So. I think that's everything I want to say. Last call on questions. Okay, then I'm going to go ahead and get started here. And this is, all this content is found in the content section of eLearn. This is this is under Unit 1, or I have it broken out into 4 units. Unit 1 is all about the microscope and cell structures and things like that. What we're going to do today is I'm going to go over the parts of the microscope, and I'm going to talk about one of the sets of tissues in the human body. Next week, we're going to do an experiment and look at the movement of different types of particles through the membrane of an artificial cell. And while that is cooking, more or less, we'll go ahead and take the microscopes out and make what we call a wet mount. You'll actually examine your own cheek cells with the wet mount. And so, because we will cover the tissue, I'll have time to cover the other tissue types, talk about mitosis in that intervening time. And so while that what we call the diffusion and osmosis lab is running, we'll fill in the time with other stuff. So we'll still get done in about an hour and a half. So even though the schedule says 2 hours and 45 minutes for the lab, we will usually get done in about an hour to an hour and a half, depending on how much we have to cover. So the time, the remaining time is yours if you want to stay here and review stuff. If you want to, particularly when we get into bones and muscles, if you want to come in and look at the bones, look at the muscles, that's fine. So when we do get into the bone section and we get into the muscle section, I will usually take one of the carts we have up here, load all the bones on it, and park it out in the hallway so you can sit out there and review the bones, review the muscles, however you want to do it, and just leave it out there all day. So, and it's fine. And then our campus police are really nice about putting the cart back in the building at the end, in here at the end of the day for us. So we used to say, just go to campus police, they'll let you in the lab. It's just easier, leave the buggy out in the hall. Who's going to steal a bone? I mean, really, I shouldn't say that. Somebody may take it as a challenge. So, okay, let's talk about the microscope a little bit here. Can everybody on Zoom see the slide that says week 1 lab? I hope. So. Good. Okay, thank you. So, because we did have a glitch the other day, I think on Tuesday. So we're looking at the microscope, and let me bring this up. There we go. Okay. Now we are using what is known as a bright field compound microscope. Now there's other types of microscopes out there. There's one called phase contrast that lets you see inside the cells and show the different structures in the cell. We're not using that. We don't have one of those. Electron microscopes are amazing. They can allow you to magnify things hundreds of thousands of times. UT has one. They probably have several. We don't have any here. The electron microscope costs, somewhere north of what we paid for the individual anatomics. These go about for 100,000 each. Electron microscopes are probably much more than that. But they can magnify things well over 100,000 times, maybe 200,000 times. If you've ever seen a picture somewhere, like online, you see a picture of they show like the pictures of bed bugs, and it looks like a dinosaur. It's in great detail. You can see every structure there. The thing's practically microscopic, and yet it looks like something from a prehistoric age. That's the resolution you get with an electron microscope. We can even see large atoms. Not very well, they're still pretty fuzzy, but we can still see atoms with an electron microscope. We don't have any need for that in this class. We're not going to be looking at atoms. We're going to be looking at cells. And we may even, at some point, we might even have looked at some bacteria. In micro, you will definitely be using the microscope to look at bacteria. But we can't see viruses. Viruses are so tiny, it doesn't really matter. We can't see them. Electron microscope can see them. You've seen the pictures of the COVID virus, the COVID virus is a spike sticking out. Well, that's electron microscope gets that picture. The Ebola virus, it looks like a shepherd's crook. You've seen that maybe somewhere. And just recently, they've spotted the polio viruses again in the sewage in New York City, which is sort of scary. Because there was a time when everybody was vaccinated against polio. Now that stopped because polio is supposed to be pretty much gone. So the polio virus, but for a case to show up in New York City is pretty surprising. The health departments routinely monitor the sewage. It's got to be a great job, What are you? I'm the sample collector for sewage. They monitor sewage for viruses. That's how they would track the cases of COVID. They could predict the cases of COVID in a city by seeing how much is showing up in the sewage. If the sewage numbers went up, then about a week later, the cases of COVID would go up in the population. The same thing would apply with polio or monkeypox, the other new one too. That's floating around out there. If it shows up in the sewage, it'll start showing up in the human population. So it's a way that can be used to track and get an estimate ahead of, to be ahead of the curve, as they say. So it used to be, hang on a second. That's okay. Glad you're here. So now it used to be that one way that the public health departments could track influenza was to track Google, because they would find that if there was a spike in searches on Google for influenza, then about a week later, there would be a corresponding spike in the number of cases in the community where the Google spike took place. So people were searching for the symptoms of influenza, and then a week later, the cases in the population would spike. And it was a very good predictor. It may still be a very good predictor of influenza. So, but it was, who would think that? But yeah, so now there's more direct monitoring. And apparently raw sewage is a great way to do that. You don't need to use a microscope for that. So now, what do we use the microscope for? Well, we can look at bacteria. We can look at cells. We can look at tissues. We can look at healthy cells, and we can look at cells that are infected. We can look at cells that may or may not be cancerous. We can examine a cell to see if there's been a change to it when it's doing something it's not supposed to be doing. Cancer cells look different. I'm not a pathologist, but I can look at a cell and say, compare it to a healthy cell and say, well, look, its shape has changed. It's growing rapidly. There's lots of cells piled on top of each other. Cancer cells don't believe in contact inhibition. I was talking in lecture about how human cells have what's known as contact inhibition. They like to keep their space. They don't like to be too close together. And I'm just looking here, and everybody sort of, has themselves positioned, with a little space between them. The ones over there like a lot of space. They're antisocial, but it's okay. No, I'm not picky.
No, you're fine.
Okay.
I just thought it was funny because I was sitting in that front row between people and now I'm over here.
Yeah, I know, but it's because we all like our space. Well, cells do too. Cancer cells don't care. Cancer cells grow on top of each other. They don't have contact inhibition. You can look at a series of cancer cells under the microscope and say, oh, they're likely cancer, malignant, because of the way they grow over. or you can look at cells and see if they're infected. You can look and see if they're growing properly. And that's where, you know, the structures come into play. We can look at cells and say, what kind of tissue is this? We can say it's a muscle cell. What kind of muscle cell? Well, there's only 3 types of muscle cells in the body: cardiac, smooth muscle, or skeletal. And you can look at it and say, ah, I know exactly what that is because each one of them is distinct. We can identify all the different cell types and tissue types in our bodies by how the cells look. And that's what the microscope comes in for us. Now, microscopes are pretty simple. They're essentially just a tube with a couple lenses in it, one at each end. The earliest microscopes were nothing more than a brass tube with a lens at the top and a lens at the bottom. And you couldn't focus it, you couldn't do anything. You had it, if you were lucky, it was on a little stand, and you stood over it, looked at it like that, and you'd have a candle in front of you. Well, this is before my time, so you'd have way before. You'd have a candle in front of you, shining giving you some light. That was 400 years ago. So they've been around for a long time. In fact, the term cell goes back about 400 years. One of the early anatomists was looking at plant cells. They didn't call them plant cells. He had a sample of cork wood, and he looked at it and he saw all these structures in there that were lined up next to each other like little boxes. He said, that looks just like the cells that monks live in a monastery, because in a monastery, the monk would have an 8 by 10 room they lived in when they weren't doing monk stuff, whatever monks do, you know. And he said, that looks like the cells, and the term stuck. So we can refer to cells based on that description from 400 years ago, because it fits, you know, because plant cells are sort of rectangular and sort of boxy looking. Now, human cells are squishy and have all different shapes. But that's where the term cell came from. So these early anatomists would use a microscope that was just a tube and look into it. Now later, over time, we've added the ability to add a built-in light source. Believe it or not, that's only happened in the past 60 years. The ability to add multiple lenses probably happened in the past 100 years. One of the great breakthroughs in microscopic science was to take the microscope and not have it stand straight up and down anymore, but to actually have a hinge on it so you could tilt it back. And then we get this, where we actually have a body tube that sticks out at an angle to make it very convenient to use. When you get into microbiology, the microscopes there have 2 eyepieces, which is very easy, very convenient, because I can tell you, if you have 2 eyepieces versus 1, you don't have headaches. So if you spend lots of time on a microscope, there's nothing like that headache you get from staring into something like this for a long period of time. Anyway, the microscope is called a compound light microscope because it has multiple lenses. That's the compound part. And it's light because it has a light source. And so the light source, here we have electric light built into it, which is great. You know, even my microscope as a kid had an electric light built into it. Yeah, I had a microscope when I was a kid. Are you surprised? You know, okay, thank you. In fact, most of us in natural sciences had stuff like that. So Dr. Smith probably had a chemistry set when he was a kid. So anyway, but prior to that, they would have prior to the electric light built in, there were mirrors, and you would shine, they would have a light source that you would shine the light onto the mirror. And that's how you got the light into the microscope. So what we have now, we're at the, you know, we have a pretty sophisticated device here that doesn't cost a lot of money for us to look at the slides and stuff on the, you know, and specimens that we want to see. We can increase the cost of the microscope and make it more precise. For the purposes of A&P, these are adequate for our needs. When you go into micro, if you go into micro, you'll have a much better microscope. It gives you a lot more clarity and resolution because you're looking at different things. Now, so the first thing that we see here is the ocular. This is the eyepiece. See if I can stop this and get on to the, okay. This is the ocular. It is the eyepiece. It is, there we go. And it fits into this, into the body tube here. And there's a lens right inside here. And every ocular has a number 10 on it, even though it's backwards in the image over there's a number 10, which means that it multiplies, it magnifies 10 times. Let me get that camera situated a little bit better. Trying to do this from two different directions. There we go. Now, so this is a lens inside here, and it says 10x on there. So every ocular will magnify 10 times. So that's an important part of the microscope. The other part of the microscope that's important as far as viewing things are what are known as the objective lenses. And the objective lenses are down here. These are the objective lenses. See right here. In this case, there are 4 of them. Each one has a different magnification attached to it based on the type of lens it's inside of it. And so they're actually, it's a standard value. Like the smallest of these has a 4 on it and a red band. They all have the same color coding. It says 4x. So your total magnification of any, for the microscope is going to be the magnification of the objective lens multiplied by the magnification of the ocular. Ocular is always 10. In this case, on the red one, it's 4. Your total magnification is 40. The yellow lens has a 10 on it. And so 10 times 10 is 100. So your low power lens is 100, gives you 100 magnifications. Your high power is 40. That gives you 400 magnifications. And your oil immersion lens, which we don't use, but you would in micro, has a 100, which gives you 1,000 magnifications. So the most powerful magnification we can get out of these is about 1,000. For our purposes, we can see everything we need to see in human anatomy at 400 magnifications. Anything over 1,000 is anything over 400 is probably getting us in too close. But if you're looking at bacteria, 1,000 is probably what you need. So, but these aren't bad little scopes. And most microscopes are going to be organized along the same lines as these. They all have objective lenses here. They all have an ocular. The magnifying powers of the lenses are going to be exactly the same. You look at, we have 2 different types of microscopes here. The microscopes are a third type. They all have the same powers on the oculars and the same color bands. Yeah, so there's some variations on, yes, ma'am. Oh, 400 is blue and 1,000 is white. So yes, 400 is blue and 1,000 is white. So and they're consistent on that. So every scope is going to have the same markings. So now let's get back to my slides. Okay, now. Okay, the light rays, light comes up from the light source here, it goes through this opening in the what we call the stage, and the stage holds a slide. And now there's a device called the iris diaphragm, and the iris diaphragm works a lot like the iris in our eye, the pigmented portion of our eye. That's what determines the shape of our pupil. It regulates the amount of light coming in. We have the same type of device here that allows us to control how much light is coming into the slide. Sometimes we want to have a lot of light. And sometimes we only want to have a little light. Now, if you, let me, well, bring this back up. Okay, now if you look at this, you can see this is the iris diaphragm. And the iris diaphragm is this structure right in the middle there. Now, if I pull this lever down, it will, I pull this lever down, it will open up like that, just like the pupil, just like the iris in our eye. If we want to rate, if we have it wide open like that, we'll let in a lot of light. If I close it, we'll regulate how much light I want to have coming in and out. So that's one way to control the amount of light we use. You know, the thing to remember is if some is good, more is not always better. You know, sometimes it is, sometimes it isn't. Sometimes you want to look at something with a small amount of light, because if it's too bright, it's hard. It makes it hard to see. Most of our body tissues, unless we have a stain or a dye on them, are pretty much transparent. When we're talking about making a section that we can view under the microscope, when we're slicing through a body part to examine under the microscope, we have to make these less than paper thin cuts. And it's, the tissues are pretty much transparent. And so a lot of light shining on that sample may just wash everything out. So we try to control the amount of light, and we use the diaphragm as one of the mechanisms for doing that. And it works. So, and it usually fits in here underneath the structure. It comes in up underneath here so that, and there's a little lever there you can turn. And I showed you how that works to control the amount of light. Now let's get back to, okay. So that's the iris diaphragm. And now I've already mentioned the lenses, but this summarizes it here. The scanning lens is the 4X with the red band. So the 4X and the 10X, so the ocular gives you 40. Low power has a 10X on it that has the yellow band that gives you 100 magnifications. High power has a 40. It's the blue band. 40 times 10 is 400. And then the oil immersion is a white band and gives you up to 1,000 magnifications. So it's 100. The reason we would use oil immersion is that if we try to magnify it 1,000 times, the light rays striking the slide actually are going to distort the images. So we put a drop of oil on the slide and lower the lens into a droplet of oil. Then we eliminate all those light rays that are bouncing around here and distorting the picture. Because now we create a uniform substance, uniform medium for the light to go through. And it works. It's messy, but it works. Okay. Now I also showed you the iris diaphragm. This is a typical compound light microscope. These are the types of microscopes they have in Morristown. I started using these slides when I came here in 2004, and they're still using these microscopes up there. So what can I say? Okay, the eyepiece. The eyepiece comes out, and in most cases, there's a little wire inside there that you use as a pointer. Some of them have them, some of them don't. You'll get a chance to play with this next week. The body tube, is designed simply to bring the light ray up from the prism. There's a big prism in this box here that sits on top of the microscope. You can see it. It's a section. Okay, it doesn't label it there, but there's a prism to bend the light rays in this box just below the body tube, so that the light can come up from the objective lens, be bent into the body tube so you can see it. So it's for something that's not very expensive, it's a pretty sophisticated piece of technology. Now the nose piece is what holds the objective lenses in place. The nose piece rotates around like that, so you can easily select The problem is, of course, if you're looking at something under low power and you switch to high power, it's probably going to be out of focus, and it's going to be the image that you're looking at probably going to be moved off the field you're viewing. And there are the objective lenses. This one only has 3 on it. So we have scanning, low, and high. And you can see the color codes on there too for 4, 10, and 40. This is the mechanical stage. The mechanical stage is another big improvement that came along about 50 years ago. The stage allows you to move the slide back and forth or up and down, for example, using these controls on the side, on the other side. Here, let's show you. I use these controls here on the side, right there. If you watch the stage, I can have the stage go front to back or side to side. Because we put the microscope slide in that big clip there and hold it in place. And then we don't have to touch anything, touch the slide to move it around. We also can move it up or down this way. In fact, that's the whole basis of focusing the slide, is by using going up or down. That's how, you know, the focus controls That's okay. That's okay. It's physiology. So, okay. Now this microscope's a little older. It has a separate dial for the coarse adjustment. On these, the large dial is for the coarse adjustment, and the smaller dial is the fine adjustment or fine focus. There's our light source. This is the base. The base is a very important structure because when you carry a microscope, you don't carry it like this and swing it around while you walk. That's not a good thing. You notice I don't have the camera on me while I'm doing that. You carry it like this. So it's just, if you're carrying it right, it's very hard to drop it. Doesn't say it can't happen, but it's just a whole lot easier to carry it because they're awkward. You know, they don't weigh that much, but they are awkward. You know, so, and I know the natural science faculty will all deny it, but we all, you know, when we're trying to put stuff away, we all carry 2 micro, we've all done it, but we won't. So everybody's looking at their phone to see whose phone's going off. But yeah, the best way is to carry it with one hand underneath the base and one on the arm. So, and while the light source doesn't seem to be a big significant piece of information, we have 3 different types of microscopes here in this campus, and every one of them has the light switch is in a different spot. So when you get a microscope next week to play with, make sure you check and see where the switch is to turn it on. You would be surprised at the number of people that I have seen over the years. My microscope doesn't work. I can't see anything. Is it on? I think. And then you go over and throw the switch and it's on. So it's very embarrassing, you know, so, but it happens, it shouldn't be embarrassing because nobody here is an expert on microscopes that we're aware of. If you are, that's great. You can show other people how to do it. And I welcome that. But if you've never used one before, you used one in high school or somewhere else and you've forgotten, there's nothing wrong with not figuring out not figuring out how to turn it on. If you've never done it before, you sit down and it's like, well, what do I do now? So. And so the light switch is important. Some are in the back, some are in the side. The dimmer switch on some of them are in the back and some are on the side also, because we do have a dimmer switch. So, okay. There's the iris diaphragm again. Now, if you're looking up close here, you can see the mechanical stage again in this one. It's not as nice as the one we have here. And the controls are on the opposite side, but they all do the same thing. They all let you move front to back or side to side. And then the focusing controls let you go up or down. Of course, there's the iris diaphragm again. Now, this is what the diaphragm also controls access to what's known as the condenser. The condenser is that little round thing right there. That is essentially like a magnifying lens. And the light rays come up through the iris diaphragm, hit the condenser, and they are focused on the opening of the objective lens. There's a little tiny opening on the objective lens the light has to go into. And we don't want to have light shining out like a flashlight, just or a headlight on a car where it just spreads out. We want the condenser brings it down to a point, and that goes right into the objective lens. So we get the most light we can focused into there. And we can, while we can move the slide around, the slide will always stay on top of the condenser, because there's the hole there in the stage is where the condenser comes through. And of course, there's the controls, for front and back and side to side of the stage. And the condenser is up inside here. There's the iris diaphragm. And yes, those things drop right off. If you want to, if you're not careful and you're trying to tighten it up in place to hold the iris diaphragm, there's a series of screws here that don't seem to have any rhyme or reason how they work. But if you unscrew them or turn them, if you think you're tightening them and they really aren't, this will drop out and land on your desk. And that's always really exciting. It doesn't hurt anything, but it's always really exciting. My microscope just broke. Well, it didn't break, but it just fell apart. So it happens, so don't, I shouldn't say it, but there isn't much you can do to hurt these things,
Challenge accepted.
There you go, what usually happens is light bulbs burn out or it blows a fuse. So then we fix it. Okay. Now, this is a good chart to remember. I've been talking a lot about the magnifications, and you would be amazed how many people get this wrong. I'll just throw that out as another challenge for you. Lots of people can't remember the magnifications. 40 times 10 is 4,000 sometimes. 10 times 10 is 10, because it's a multiple choice test and you read it, you don't see the right answer and you jump on it real quick and you don't think. So just this slide is a good little study tool just to remember the magnifications. So I'm not criticizing, making fun of anybody. I'm just speaking from years of experience doing this and seeing the mistakes people make, that they're kicking themselves later for that mistake. We've all done stuff like that. We've all put down the wrong answer. We've all driven through red lights that we thought were yellow, right? So there, yes, thank you. Yes, I'm right there with you. So, okay, resolving power. This is something else that I'll show you an example here in a little bit of how this really helps us. Resolving power is the ability to distinguish between two objects, to look at fine detail. If I were to stand outside, we were all outside, and I stood at one end of the parking lot and held up these two pencils like this. Everybody could probably see that I was holding something, my wife is 2200, so she's like, "Are you holding it up yet?" So uncorrected. So now resolving power, this far apart, you have no trouble. But if I'm still at the end of the parking lot, I'm holding the two pencils like that. Can you tell it's one or two? Now here you can tell it's two, obviously. But when you get 100 feet or 200 feet or 300 feet away, you can't tell there's two. Our eyeballs don't have the ability to differentiate between one image or two images. Our lenses and our eyes aren't that good, but suppose you had a pair of binoculars and you could look up at what I was holding up and say, yes, there's two pencils, not one. That's what the microscope does. structures that we would see with our own eye look like it's very precise or smooth. We don't see the irregularities. We don't see the edges to the image. We don't see any of that stuff because our eyes can't see the irregularities. Now, this is probably the best example. This is a letter E. It's upside down and backwards. Forget that part for a second. That is a letter E like you would see on a printout. You know, like if you had something I print like something on the schedule off of a laser printer. You know, you look at it and it's 12 point or 11 point or whatever I used, and it looks smooth and crisp and clean, right? You look at it and say, oh, it looks great. Except this is what it really looks like magnified 100 times. What you see there are the irregular edges caused by the toner being slapped onto the paper, or maybe the ink being splattered onto the paper. And so instead of a nice crisp image, we see a splatter pattern. And yet when we look at it on paper at 12 point, we can't see any of that. Our eyes aren't good enough to see those irregularities, but the microscope shows it to us. This is resolving power. Microscopes let us see great detail that we couldn't see otherwise. And there's a point to this. I'll bring it up here when we look at epithelial cells. So this is resolution. The other thing is inversion, upside down and backwards. These images that we see in the microscope are all upside down and backwards, but it doesn't matter. The cells don't care. You know, a cell doesn't have up or down, or very rarely doesn't have up or down. And so whether we see it this way or see it the other way, you know, on letter E, we can see the difference. You look at a cell, you can't tell. If you've ever looked at the moon with a telescope, you'll notice that the image is usually backwards on the telescope and what you're seeing. If you take pictures through your telescope, you gotta usually flip the image to get it looking the right way. So yeah, I'm a geek. I do that kind of stuff too. So, but you know, now the moon, the moon, you can really tell the difference because you can see this. You can see the seas and some of the big structures with your own eye, but you have to flip it to see it how we view it here. Okay, enough of that. Now I mentioned stains. We have to stain tissues when we're going to examine them because most of our tissues that we look at have no color. And so without color, there's no contrast. We can't tell what it looks like. There are very few structures in our bodies that have any kind of distinctive color. The gallbladder is green simply because of the presence of the bile in there. The liver is dark red, and some of the muscles are dark red, but not very many. And so when you cut them down to the level of one cell layer thick, when you're looking at them to make a slide, they're pretty much transparent. So we use a stain, some sort of dye, to give it contrast. And one of the best examples of a dye that we're going to do this next week. You're going to take a toothpick and you're going to scrape the inside of your cheek with a toothpick and smear that on a microscope slide and put a drop of methylene blue on that smear, and then put a cover slip on there, and you're going to see cells. And the methylene blue will act as a dye. It'll look like this. Human cheek cells, and they're dead. They've been dead for probably a day, and they're dying all the time. And the living cells underneath are pushing up the dead ones, and they just slide off. They're there to be worn away to protect the inside of our mouth. The cytoplasm, that's just the goopy stuff inside the cell membrane, and the nucleus, that's the big dark blue spot there, absorb the dye in different concentrations. So we can see three things. We can see the nucleus, we can see the edge of the cell at the cell membrane, and we can see the cytoplasm. And every nucleus in our bodies is exactly the same. The DNA that you scrape off a cheek cell and you look at it, and under the microscope, and it'll look something like this. This is what a real one, this is what one of our homemade cheek cell slides look like. Everybody will have a DNA, have a nucleus in their cell, and the nucleus has their DNA, and all the DNA in that cell is the same as all the DNA in all the other cells. The only reason it's a cheek cell is because all the other information is turned off. I'm a cheek cell, so I'm not worried about livers or bones or muscles or things like that. If I'm a liver cell, I'm not worried about cheek cells, but the DNA is there. It's just not being used. So you'll see your nucleus, and we know that that's where your DNA is located. And sometimes you get a lot of cells. Sometimes it works very well. So, and we'll do this next week. It's a very easy but interesting little exercise. Plus, it'll give you some time to play with the microscope. Now, I want to go to the epithelial tissues, which won't take us very long to go through. Yes, ma'am.
When we're looking at the cells,
Yes.
will we have the availability to possibly see bacteria as well, or is that?
We might. You know, it depends on the dye we have and if the bacteria is present. Now, if you're scraping the inside of your mouth, there's probably lots of bacteria. Whether, I'll ask Dr. Malley what dye we could add to see if any bacteria would show up. Because we can run it up to 400 magnifications.
Yeah.
So I'll ask him about that. So now let's take a look quickly. Here we go. Tissue types. Epithelial tissue. I need to go back into sharing. There we go. Now, there are only 4 tissue types in the whole human body: epithelial cells, muscle cells, connective tissue, and nervous tissue. Epithelial cells cover the outside of the body. They line every opening into the body. That's what they do. Every entrance to the body, every exit to the body. The digestive system is lined with epithelial cells from start to finish, all 26 feet of it. The skin is covered with epithelial cells, as is the urethra, the uterus, the vagina, the cervix, the ear canal, the optic cavity behind the eyeball, the nasal cavity. All these are covered with epithelial cells because everything, all that is exposed to the outside. And so epithelial cells are protective cells, are surface cells. The other 3 types are muscle, connective, and nervous. Now, if you are, and we'll get to those next week, if you are an epithelial cell, there's only 2 categories. They're either simple, 1 cell layer thick, or stratified, multiple layers. And most epithelial cells come in single only. So they're simple. And there's only 3 types, 3 big categories. 3 big types within the categories. If they're simple or stratified, they're either squamous. A squamous cell is an irregular shaped cell. It's flat and has a big nucleus in it that sticks up and it looks like a fried egg. Those cheek cells that you saw a few minutes ago, those were squamous epithelial cells. The nucleus is actually 3-dimensional. Can't tell from looking at it, but it bulges up and down in there. We find that squamous epithelial cells, we find simple squamous forming the structures in our lungs. We have the alveoli, our areas for gas exchange in our lungs are made of simple squamous epithelial cells, 1 cell layer thick, the consistency of wet toilet paper, single ply. We have about 1 million alveoli in each lung for gas exchange. Bless you. The alveoli are surrounded by hundreds of blood vessels. So we have great gas exchange. Oxygen comes into the alveoli moves into the blood vessel, CO2 leaves the blood vessel and goes into the alveoli, and then we have gas exchange. So simple squamous epithelial. Now we could have stratified squamous. In fact, that's what you're going to scrape off the inside of your cheek. Stratified squamous is multiple layers. It's what we have in our skin. That's why we don't bleed constantly when we bump into things, because we're scraping off layers of multiple layers of stratified squamous cells. When we were kids and we fell down all the time and hurt our knees and our elbows and bled all over the place, and those were stratified squamous cells coming off. Yeah. To get to the blood, you got to go through all the layers of the epidermis and get into the dermis. So, because think about all the times when we run into things or fall down and trip, we don't bleed. Yeah, we only bleed if we penetrate through the stratified squamous. Second type of epithelium are cuboidal cells. They're called that because they're shaped like they really are shaped like a cube. We find these primarily in our kidneys. They're stacked on top of each other to form tubes. And that's how we filter all the waste products through our kidneys constantly. Columnar cells are shaped like columns. And we use columnar cells when we want to, we use simple columnar cells when we want to absorb things. We absorb all of our nutrients in the small intestine. Everything we eat and drink is absorbed in the small intestine after it's been processed through digestion. And the small intestine is lined, all 20 feet of it is lined with millions of columnar cells. 1 cell layer thick, nutrients are absorbed by the simple columnar cells, and then they pass through them and into a blood vessel, into a capillary right behind the epithelial cells. And then all the nutrients then from the small intestine are taken to our liver, where we clean it up and detoxify it, and then we send it into our blood supply. And we have 2 sort of outliers. We have transitional. Transitional epithelial cells are cells that look like columnar cells, but then they stretch out and look like squamous cells. Our urinary bladder is made of transitional epithelium, because our urinary bladder can go from something the size of a walnut to something the size of a grapefruit. It gets pretty uncomfortable when it's that big. Because that means it's holding a size of a walnut, it's holding about 100 mils of urine. Size of a grapefruit is holding a liter. And a liter is really making itself, telling us, hey, that, I really got to go. But the bladder can stretch. It can stretch out, and then it can shrink back. When it's small, it looks like it's made up of columnar epithelial cells because it's open to the outside. When it stretches out, it looks like it's made of squamous cells. And then the last category, oh, thank you. I thought you had to go again.
Yeah.
Okay, so the last category is the pseudostratified ciliated columnar epithelial cells. Pseudo means false. They look like they're stratified. They're ciliated. They look like they have little hairs on them. They're columnar. They have columns. And they're pretty unique. So anyway, this is simple squamous. This looks, the stratified squamous and the simple squamous look exactly the same. Big nucleus, which actually bulges up and down, an irregular flat shape. Yes.
Would the urinary bladder and the stomach have the same type of tissue then, since if that stretches, the stomach can also stretch?
Yes, the stomach is also made up of epithelial cells too. And because the stomach can stretch, there's probably transitional epithelial in there too. So, and the stomach also has a lot of mucus generating cells in there too, because of the acids. So, okay, these are the cuboidal cells. These, what you're looking at here, you're looking straight down into a kidney tube. Sort of like looking down into a pipe. Imagine, those things are pipes looking up at you. And our blood plasma is filtered through our kidneys. As I said, we run our entire blood supply through the kidneys 60 times a day, and the filtrate that's generated, the fluid portion is in these tubes, and the material that we want to recover is absorbed on either side, and the waste product stays in here. We generate, and you'll hear all about that in AP2, so don't worry about how that works. But we generate about a 1.5 liters of urine every day. And the urine is what's in these tubes here as it works its way through the kidney. Simple cuboidal epithelium. Here's a really nice shot of what they look like. Definitely have a cube shape. And again, you're looking straight down into a kidney tubule. These are the simple columnar cells that we find lining the small intestine. In fact, this is a portion of the small intestine. And you can see they line up like little columns right there. But what makes them unique in the small intestine are these cells. These are called goblet cells. Now, the goblet cells didn't take the stain as well as the columnar cells did. So the goblet cells are pretty pale. They are secretory in nature, and the columnar cells are designed to absorb. So nutrients go into the columnar cells. The goblet cell secretes mucus to lubricate the passage of food and any other material down through the digestive system, because we don't like friction. We start lubricating food in our mouth as soon as we start eating it. We lubricate the inside of the esophagus. We generate a lot of mucus inside the stomach and the entire length of the small intestine and into the large intestine too. We use goblet cells everywhere. We want to have mucus at our rectum and anus because we don't want to have it hurt when we defecate. We lubricate the vagina and the uterus and the cervix during intercourse and during childbirth. We lubricate the inside of our nasal cavity, not to make the passage of air better, but to allow the air to become moistened. The goblet cells in our nasal cavity release mucus so that the air can become moist, because moist air is easier to inhale. It's also cleaner too. So these goblet cells have a real role to play here. We see them here. We see them on this one. This is still the cross section of the small intestine with the goblet cells there. All these pale structures are goblet cells. You can really see the column shape here. And back in here, these are all capillaries back here that are going to suck up the nutrients that have come across. Now this is the pseudostratified ciliated columnar epithelial cells. What you see here, the reason we call it pseudostratified is it looks like these columnar cells are stacked on top of each other, but they're not. They just look like they are. Now, a couple of things are very visible here. First of all, these little things here are cilia. Now, you can't see those at 40 magnifications. You can't see them at 100 magnifications. You have to get up to 400 before you can see them, because cilia are extremely tiny. But what they do is, if you'll notice that these are goblet cells here. These are all goblet cells there and there and there and there and down along here. We see pseudostratified ciliated columnar epithelial cells lining our airway, our trachea. And they're coated with the mucus that those goblet cells generate. So when we suck in air, any dust and dirt and debris and pathogens that are still there after it came through the nose are trapped in that mucus. And then the cilia move. The cilia move in one direction. They beat uphill. They're constantly moving in an uphill direction, push that mucus up into our throat so we can swallow it. We swallow about a pint of it a day. We're not aware of it, but it happens. It's
yummy.
Yeah, I know, but thank goodness we don't think about it. So, but that's what the cilia do. They push this mucus along. If you have a patient that's a smoker, tobacco smoke paralyzes the cilia. And so your patient that's a smoker will usually start the day every day coughing out the mucus or coughing up the mucus because the mucus isn't moving. They have to cough up the trapped particles and dust and dirt and debris. The term smoker's cough comes from this mucus they have to get up. The good news is they stop smoking. Within a few months, the cilia become active again and it works. So, but for some reason, the cilia just, that smoke is an irritant and they stop moving. We all have this cilia in our airways. Another location that the cilia is found is in, there's the cilia, is in the fallopian tube. Now you'll find when we get into AP2 term, we use fallopian tube, which we've used for forever. But now we also call it the oviduct or the uterine tube. All three of them are used interchangeably. Most people are going to still say fallopian tube. But the uterine tube, the fallopian tube, the oviduct is lined with pseudostratified ciliated columnar epithelial cells because once an egg makes its way into the fallopian tube, it has no way to get to the uterus. The egg is a little tiny round ball, and whether or not it gets fertilized is up to whether or not there's sperm present and the timing is right. But it still isn't going to help the egg get to the uterus. So the tube is lined with cilia that will help to push that egg along. It takes 7 days for that egg to go 2.5 inches to get to the uterus. The egg is ovulated, always ovulated on day 14 of the ovarian cycle, because whatever day ovulation takes place, the count starts as day 14, and it's got 14 days before menstruation starts. It has to be implanted in 7 days if it's going to implant. And the only way to get there is for the cilia to push it along to get down to the uterus. And the cilia look something like this. Again, they're going to move in one direction. Here they're designed to sort of shove and push and drag the egg along. And it takes a while, to get a microscopic structure. If it's fertilized and it's growing, you still have to push that thing along, 2.5 inches. It takes about a week to get there. And whether it's fertilized or not doesn't really matter as far as it's being moved. Now, in some cases, it may not make it. In some cases, it gets stuck and then degenerates. In some cases, in very rare cases, a fertilized egg may get stuck in the fallopian tube and start to grow. And then we have some other complications with that. So, okay, and this is, we're just about done. This is the urinary bladder. The lumen is the open part of the bladder. What we're looking at is the wall of the bladder. This is a bladder that is relaxed. It's empty. Because, how do I know that? Because what we're seeing here are what looks like columnar cells along the wall of the bladder. Now, when it fills up, it will stretch out and flatten out, and it will look like squamous. But right now, if the bladder is empty, then you're going to see it looking like these columnar cells. This is why we call it transitional, because it goes from one shape to another shape. And where we see that, we see it in the ureters coming from the kidneys. We see it in the urinary bladder. We see it in a little bit the top portion of the urethra. We probably see it in other organs that stretch too, like in the stomach, for example. Anything that would stretch may have something like a little bit of something like that. But primarily, we're going to see it in the urinary bladder, because the urinary bladder, as I said, can go from 100 mils up to a liter. We're uncomfortable at a liter, but it'll stretch that much. So, and this is what stratified squamous looks like. Lots and lots and lots of layers of usually dead cells. Stratified squamous epithelium. This is what lines the inside of our cheek. This is what lines our rectum and our anus. This is what's lining our esophagus. Anywhere where we're going to have wear and tear, lines the vagina, and the uterus and the cervix. It'll, wherever we want to have protection from wear and tear, we're going to have multiple layers here. All of our skin is stratified squamous. Some areas are dry and mucus-free, like our skin. Other areas are lubricated extensively. If we don't want to have friction, we have lots of goblet cells and lots of mucus, because our bodies do not like friction. If you think friction doesn't have a lot to play, take your hands and rub them together for just a couple seconds and feel how hot they get. I mean, that's the friction. So, and that, I think, oh, there's another picture of stratified squamous. So you can see how that we have the multiple layers there. Okay, and that is the end of epithelial cells. So anybody have any questions on any of this? Yes.
So transitional and stratified, are they not in the two categories? Just like
They're sort of outliers. They're not part, we don't consider them part of the three big categories, simple, squamous, cuboid, or columnar. They're, because they only show up in just one or two places. So we keep them as a separate category. Or a separate, yeah, I guess we'd say that.
Exceptions to every.
Right, they're the exceptions, they're the outliers, or the ringers, if you will, so. Okay. So if there aren't any questions, we can call it a day. And those of you that are Zooming, thank you for coming in. We'll see you next week.